Look at glucose. This glucose is converted into glucose 6-phosphate. What is the meaning here? Glucose converted to glucose 6-phosphate. What is the change? What happened? Phosphate at the 6th carbon. That's it. So glucose is added with phosphate at the 6th carbon. That's it. You don't need to know the structure for that. No structure. As long as you understood, right? What is the meaning of glucose 6-phosphate? Phosphate at the 6th carbon of glucose. That's it. So who is going to add that? It's a phosphorylation process. You are phosphorylating glucose. So any enzyme that phosphorylate is called as kinase. Is it not? Protein kinase. Phosphorylation enzymes are called kinases. So what is phosphorylated here? Glucose is phosphorylated. That's why the name of the enzyme is glucokinase. Then what is the meaning of hexokinase? Hexokinase is glucose is an exose, meaning it has 6 carbons. As you see, there are 6 carbons in a glucose. So hexokinase is a general enzyme. Means all exoses. Glucose is not the only exose. You have mannose, galactose, fructose. These are all exoses. So that means hexokinase, it can add phosphate to all these exoses. Only thing is it may have some preference. It may have more preference to glucose, then galactose, then fructose. Otherwise it just adds phosphate to all exoses. So we have two enzymes to do the same thing here. First reaction, which is an irreversible reaction, conduct means there are two enzymes mentioned. One is hexokinase, other is glucokinase. The question is, why you have two enzymes to do the same thing? It could have been just one enzyme, maybe glucokinase or just hexokinase. Why two for one? The reason is you need to know where these enzymes are located and what is the purpose and what are the characteristics of these enzymes, and that makes them necessary to be placed in certain locations in our cells. Okay, we are going to get into that area in the next slide. Before I take you into that, like before I make you understand why do you need two enzymes to do the same thing, let's understand what is the purpose of doing this. What is the purpose of phosphorylating this glucose at the 6th carbon? Remember, you have seen digestion and absorption of carbohydrates, right? There are amylases, maltase, sucrase, isomer, all these enzymes. So what they do? They break complex carbohydrate all the way into monosaccharides and helping in absorption. Once they are absorbed in the intestine, where they go? They go to GLUT2 and then into portal system and then into blood. So now you know your glucose, which is a major monosaccharide in the blood. And remember, glucose is a polar molecule. It has hydroxyl groups. It cannot just go through the cell. It needs a glucose transporters. There are a variety of glucose transporter you have seen, right? So once glucose enters, remember these glucose transporters are bidirectional transporters. Bidirectional means work on either side. If the glucose is going in, it can also come back into the blood. Bidirectional, two-way street. You can go, you can come back. Okay, that means all glucose transporters, GLUTs, they are bidirectional. That means if you have more glucose in the blood, it gets down concentration gradient, right? It is going down into the cell. And if the cell is not using that glucose, if the glucose is building up in the cell, it will come back into the blood. Bidirectional, right? So what cell has to do if it wants glucose? It has to trap that inside. Okay, so it's like I will give Suja $100. The condition is you should use it in 24 hours, otherwise I'll take it back. I'm not that rich. So otherwise I'll take it back and give, what's your name? Amila. Milan. Okay, I will give it to her. I will give him 24 hours. If you don't use that, I'm going to take it from you and give it to her. And the condition for her are 24 hours. If you don't use it, I'll take and give it to Alexa. That's it. That's what happens in our cells. Cell will have, means blood glucose taken up by the cell. Cell has to use it. How cell will use it? Means how cell will make sure that it doesn't go back. It is simply phosphorylates it. Phosphorylate the glucose. Once it is phosphorylated, it is there in the cell. It is not going to come out because there is no glucose 6-phosphate transporter out. Glucose can come out through GLUT transporter, but not glucose 6-phosphate. That means it is committed for the cell now. If he spends $1 out of that $100 is his. That's it. That means it is committing. Phosphorylation is simply committing glucose for the cell. And that's what I written. Phosphorylation commits glucose to cellular metabolism, not necessarily glycolysis. It is simply, it is committed for the cell. It's like you right now committed for what? For MD, correct. You are committed now. There's no returning, no return. No return, that's it. If you like it, doesn't like it, you got to do it. That's how it should be. Find alternate solution. Oh, what if this is not there? I will do that. No, this is the. That means you will do it. That's how the commitment is, right? You commit, you are committed. You give under everything, right? This glucose is now committed for the cell, not necessarily for glycolysis. You are committed for MD, not necessarily for surgery. It can be obstetrics and gynecology, it can be ophthalmology, whatever it is. You are just committed to MD. Like that, glucose is committed to the cell. It can go into glycolysis, it can go into pentose phosphate pathway, it can go into glycogen synthesis, it can go into glycoprotein or proteoglycan formation. See there? So the first reaction is not a rate limiting step. It's not going to determine the rate of glycolysis. It is simply committing glucose for the metabolism. That's it. And that is the very purpose of very first reaction in glycolysis that you should know. That is why there is phosphorylation. You got this? Okay. So now let's look at glucokinase and hexokinase. Glucokinase is an isoenzyme of hexokinase, and they have a different Km and Vmax for each one of them. Both glucokinase and hexokinase differ in their tissue location, Km and Vmax for glucose. What is Km? What is the meaning of Km? Km is indicative of what in terms of affinity? Lower the Km, higher the affinity. That means if the enzyme has low Km for something, it is able to work even at low concentration because it has higher affinity, right? So higher the Km, lower the affinity. That's one thing. Second thing, what is the information about Vmax? What it indicates? Vmax is the capacity, right? If the enzyme which has got higher Vmax, that means it is able to take as much substrate as possible because it has high Vmax capacity. Low Vmax means it will saturate early, right? It's like someone eating this much food and feeling full, someone eating this much food, still not feeling full. You seen your friends? Somebody eats so less and oh, I am full. Somebody eats so much. Is there any food left? That's the capacity somebody develops. You know, that's the capacity, digestive power. Like that, our enzymes have got low Vmax and high Vmax as well. Low Vmax saturate early, even with less concentration of substrate, whereas high Vmax means it will go on. So, understanding this concept, let me take you into these two enzymes. It is important to know this table. First, you understand. Later, it will be easy for you to remember. Hexokinase. Let's focus on the Km and Vmax of hexokinase. Km and Vmax. Look at hexokinase. What is its Km and what is its Vmax? Low. Hexokinase has low Km and low Vmax. What it means? What is the meaning of this? Low Km meaning what? It has got super higher affinity for glucose. That means what? It is able to conduct a reaction even at low glucose concentration. That's what it means. right? Even if you are in hypoglycemia, less than 35 milligrams per deciliter is hypoglycemia. Even if you are in hypoglycemia, still your hexokinase is able to work because it's a high affinity enzyme. It loves glucose so much. So it's going to work. Whereas low Vmax means what? It's going to saturate early. It's going to work at low glucose concentration, but it's going to saturate early, right? So that's a low Vmax. That means it's not going to consume too much of glucose. That means it's not going to commit too much of glucose for the cell. Means hexokinase working means what? It is going to add phosphate to the 6th carbon, is it not? What is the function of hexokinase? Glucose to glucose 6-phosphate. What is the purpose of phosphorylation? Committing that glucose for the cell. So if your hexokinase is a low Vmax enzyme, that means what? It's not going to convert too much of glucose into glucose 6-phosphate for the cell. It is going to phosphorylate only that much of glucose which is necessary for the cell, not more than that. And that's what I have written down there. Cannot phosphorylate more sugars than the cell can use. That is what hexokinase will do. It will make sure even if the blood has more glucose, it will take only what is required. It's like I'll give $1,000 to Arib and your necessity is only $150. Just consider, just for one day. 150 is too much for one day. Okay, $150. And I'll give you $1,000. So what do? You take 150 and give rest to Suja. And so you have 850 left, right? And you will take 150 and then give 700 to her. And she'll take 150. Give rest 550 to her. Something like this. So that means we have limited amount of glucose in the blood. Everybody has to use it. Okay, everyone has to take it, like distribute, even distribution has to happen. Everybody has to use the same glucose. There are a lot of tissues, lot of cells. Everyone has to get their share, right? And that's what hexokinase will do. Hexokinase will make sure everybody gets their share without any partiality. And that is why it is important for hexokinase to have a low Vmax so that cell can use only what is required, rest will go back and used by the other cell. Okay, imagine that Harib has retained $850 instead of 150. He has retained 850 and gave 150 to him, and then he will retain 150. What about her? What about her? What about everybody in the class? No dollars. That means you go into hypoglycemia, is it not? Lack of cash. Everybody go hungry. Only Harib is happy. Is it not? That's what happens. That will happen if your hexokinase becomes high Vmax enzyme. If your hexokinase become high Vmax enzyme, you get into hypoglycemia. That is why it is important that hexokinase is a low Km, making sure it is providing glucose at all times. But at the same time, it should be low Vmax so that cells cannot use more glucose than what they need, so that it will prevent hypoglycemic condition. Now come to glucokinase. Look at glucokinase. Glucokinase is a high Km and high Vmax enzyme, exactly opposite to hexokinase. Now, to understand this, why it is high Km, high Vmax, you should know where is the location of the glucokinase, hepatocyte and pancreatic beta cells, liver and beta cells of pancreas. What is the function of beta cells of pancreas? Release of insulin. We know the mechanism, glucose goes in, depolarization, all that. So, release of insulin. And when you expect glucose to go into the beta cells of pancreas? When do you expect blood glucose to enter into the beta cells of pancreas? Only when there is high concentration. Only when you are in relatively hyperglycemic condition, then only beta cell has to allow glucose to go in, because once it goes in, it will bring insulin. And insulin is a hypoglycemic hormone. So the response should be with hyperglycemia. Whenever there is more glucose, insulin has to be released so that insulin comes and normalize glucose. That's how it should be. That is what our body beta cells will do. So that means beta cells should take up glucose only when there is higher concentration. That means it should express such an enzyme which has got high Km for glucose. That means low affinity for glucose. It should not work all the time. It should work only when there is a higher glucose. That is why glucokinase is a high Km enzyme. Its Km is 5 millimoles. Okay. So it is about 5 millimoles. It's about like 70 milligrams per deciliter something. Right. So that time it will allow glucose to get in. And also know that as long as glucose is high, it should be able to take it up and convert glucose into glucose 6-phosphate so that insulin is going on releasing to bring the glucose down to normal. Once it comes to normal, because it is a high Km, it stops working. Right. So that means having high Vmax will make sure that sufficient insulin is released as long as there is a high glucose available. That's one thing. Coming to the hepatocyte. What is the function of hepatocyte? Hepatocyte is like, it's like I was saying this last semester, like mother. Liver is like mother. Remember, in the metabolism, liver is like mother. You will understand this. If you really, like metabolism, you will become a really good physician. Let me tell you honestly. If you like biochemistry and physiology, you will be a fantastic physician. Like internal medicine, right? And of course, it is connected to all other specialties as well, because these are fundamentals. Once you understand the fundamentals, your foundation is strong. So you can build on that. Okay. So in metabolism, liver is like mother. What she does is, let me call liver as she. What she does? So she will eat only when there is plenty in the family. When there is no food, what mother will do? Little food available, she will share it with children, maybe later husband. First children, after that husband. It's like that, right? That's what she does. So that means liver, when there is plenty, she will eat. That means when there is plenty of glucose, only at that time she will take. Other times she won't, right? And when there is less glucose, what liver will do? She will make glucose and release into the blood. That is what is gluconeogenesis. She will burn fatty acids and give ketone bodies into the blood so that everybody uses it. And this is what liver does. So liver is taking care of all other organs in our body, their metabolic needs. The role is done by liver. That is why if you know, liver has got a highest regenerative capacity. If you remove 90% of liver and only retain 10%, it will go revert back into 100%. That's the highly regenerative organ. You know that, right? So if you remove half of the liver lobe, it will come back again, because this is the organ that will maintain our metabolism. Entire organ, fuel requirement is maintained by liver. Okay. So liver has got this glucokinase, which is a high Km and high Vmax enzyme, meaning it will work only when there is plenty. But as long as it is plenty, it is able to take it. And then what liver will do with that? Is going to convert that into glycogen, is going to convert that into fatty acids, make it as triglycerides, and send the triglycerides to adipose tissue and safely store it there. And glycogen stored in the liver. So for short-term energy needs, glycogen breaks down. For long-term energy needs, fatty acids will break down. See the thing? Is it not fantastic? Should love this. I'm saying this since 20 years. Yeah, as if I am saying for the first time. See, that's the love you should have. Then it will come. Is it not? Okay. So that is why you need a glucokinase, which is a high Km and high Vmax enzyme. See, I have written here. This allows the liver to effectively remove the flood of glucose delivered by the portal blood following a carbohydrate-rich meal, thus minimizing hyperglycemia during the absorptive period. It will prevent hyperglycemia. by taking up extra glucose from the blood. And that is why we need to have glucokinase, which is a high Km and high Vmax enzyme. Now let's see what can happen if there is a mutation in glucokinase. Let's imagine there is a mutation in glucokinase where it becomes a low Km enzyme. Low Km but high Vmax. Low Km, high Vmax. What will happen to your blood glucose? It will become low because liver is starting to take all the glucose, whether high or low, it is just taking it up. So person go into hypoglycemia. So that is why these enzyme mutations can alter our blood glucose level. This is what is the important information that you should understand from this slide. Other things you can read. Glucokinase is a glucose sensor because it will allow only glucose when it is plenty, all that. And look at the regulation down here, which we are going to go over again. Hexokinase is feedback inhibited by its end product, glucose 6-phosphate. That's a classic feedback inhibition. Whereas insulin induces glucokinase in the hepatocytes. Whenever a person is in well-fed state for a long period of time, remember, there is an induction of glucokinase. Induction means at the gene level, more and more glucokinase is produced because you are keeping yourself in a well-fed state for a long period of time. Like somebody eating a lot of carbs every day. What happens? Lot of carbs every day. What happens? We will put on weight. Our liver goes into fatty liver. Remember fatty liver? Type 1, like stage 1, stage 2, fatty liver, stage 3, go into cirrhosis. This carbohydrate, like addiction to sugar, is a bad thing. Sugar is addictive. Once you start eating, you cannot stop. Start eating sweet. You cannot stop with small little thing. You want bigger share. And then you eat, let me eat one more piece. You'll eat one more. You keep it in the fridge and you always see after meals, let me eat a little bit, and you eat. You can't control. Morning you say, I should not eat, I just control. After dinner, after dinner, let me eat little sweet. Is it not? This is an addictive thing. More sugar, more carb means higher glucose. And this glucose, our body is designed in such a way that we are going to store it for future needs. What liver will do? This glucokinase allow glucose to get in, high Vmax, making sure as long as glucose is there, it is coming in, and there is a limited storage of glycogen in our liver. Only limited glycogen we can store. Rest all is made as fatty acids and triglycerides and sent to the adipose tissue and store it there. There is no limit to it. So as long as you eat more glucose, it is all going and storing in the adipose tissue. It is all direct connection. So how do you prevent buildup of adipose tissue? Stop eating. Stop eating what? Carbohydrate, sugars, refined flour, anything that has refined, sugary thing. Stop. Then you slowly decrease your adipose tissue. This is what it is. So even if you go and spend 2 hours in the gym, if you don't control your diet, not going to work. You got to control diet. 80% is diet, 20% exercise. Ask him, see, he has good muscles. What's your name? Jose. See how you control. Exercise, you build muscles. Muscles are the major consumers of your glucose. But it doesn't stop there. You should control your diet. If he ate a lot of sugar, lot of things every day and then go and work out, he won't get that shape. No. You got to be careful with your diet. Okay. So that is what is about. What I'm trying to say here is, if you are eating high carbohydrate diet all the time, because there is so much glucose coming into the blood, liver has to use it, take it and safely store it. That means the very first enzyme which is taking care of that glucose has to be at higher concentration so that it is going to commit that glucose for the liver and put it into metabolic pathways. That is why it is induced. Remember this. So how to avoid induction? Control carbohydrates. Okay. So there is no feedback inhibition of glucose 6-phosphate over glucokinase. Product inhibition is not there. Once it is converted, that's it. It goes into fatty acid. There is no feedback. on glucokinase, whereas hexokinase that is there. All right. So that's about glucokinase, hexokinase. We'll have a break here for 10 minutes. When you come back, we will go into other reactions. Thank you. Okay. Now let's get into applied clinical aspect related with first reaction in glycolysis, and especially glucokinase reaction in beta cells of pancreas. If you refer to this figure here, which you have seen in digestion and absorption of carbohydrate lecture. All of you seen this figure? Remember this figure? This is in digestion and absorption of carbohydrates, where glucose enters into beta cells of pancreas through GLUT2 transporter, and the glucokinase in the beta cells will convert glucose into glucose 6-phosphate, rise ATP levels, and that rise in the ATP levels will go and bind to ATP sensitive potassium channel. The name of the channel is ATP sensitive potassium channel. And who is the regulator of this potassium channel? Which molecule regulates this potassium channel? ATP, if that is what is asked. That's why the name of the channel is ATP sensitive potassium channel. So potassium channel closed means there will be depolarization of membrane, and depolarization of membrane causes opening of calcium channel. Calcium enters and it will lead to release of insulin along with C-peptide. For every molecule of insulin released, there is a release of C-peptide. That's why if you want to know how much endogenously synthesized insulin is produced, all you can measure is C-peptide. You know how much insulin is made. Okay. So that's how insulin is released, and that insulin will make sure blood glucose is dropped back to the normal range by expressing GLUT4 transporter on the skeletal muscle, cardiac tissue, adipose tissue, all that. Okay. Now, what the very first enzyme in this process where it is handling glucose, incoming glucose, is this glucokinase. What if this enzyme is mutated? Mutation in the gene coding for pancreatic glucokinase, it will lead to drop in the insulin release, is it not? If you don't conduct glucokinase very first reaction, all these things don't go on. That means there will be drop in the insulin. Insulin won't respond to rise in the blood glucose level. So that is what happens in a disease called maturity onset diabetes of the young type 2, MODY2. The mutation is in pancreatic glucokinase. Make sure that is your answer. Okay. It is a type of diabetes. So as you can expect, one of the key sign of diabetes is what? Hyperglycemia, excess glucose. Patients will show hyperglycemia and it will appear in the urine. So there will be glucose in the urine, all that. So hyperglycemia signs you can see in these patients. What are the signs of hyperglycemia? That is polyuria, polydipsia, and polyphagia. That means frequent urination, and then frequent being thirsty most of the time, and also polyphagia. Polyphagia means eating frequently. They feel hungry majority of time. It's all the osmotic effect of glucose. Remember, excess glucose in the blood. I told you, carbohydrates are osmotically active molecules. Excess in the blood or excess in the urine, it will have its osmotic effect. So the osmotic effect of your glucose will lead to all these directly or indirectly. This is because of osmotic effect of glucose. Polyphagia, polydipsia, polyuria. That is how MODY2 patients may express, some of them. That is called symptomatic form of MODY2. Asymptomatic form means these patients will have hyperglycemia, but they don't show physical signs and symptoms. That is asymptomatic. And also note that this is it runs in the family. It's an inherited condition. MODY type 2 is an inherited condition. That means there will be family history. Please write it down. Family runs in the family. Let's write it that way. Runs in the family. Runs in the family. Because it is an inherited condition. Okay. So these are some of the things that you need to remember. Type 2 diabetes. Let's go to type 2 diabetes, most common one in the world. Type 2 diabetes. So what is the cause for type 2 diabetes? What is the major cause for type 2 diabetes? Obesity, overweight and obesity, right? Overweight, obesity. That means nobody runs in the family. If you run, why you will be overweight and obese? No one runs in the family. That's why you're overweight and obese. You run, you are not overweight, obese, you don't get diabetes. Is it very simple? Prevention and treatment of diabetes, prevention especially, it is very simple and easy. You don't need to pay money for that. But we don't do it. We don't do it. So that's what it is. So run. So anyway, so that's about type 2. Sorry, not type 2, MODY type 2. That is maturity onset diabetes of the young type 2.